Okay, so the recording is started. This is Bio 208 with Dr. Granier. We're in the fall semester, and today is the last day I have dedicated to topics in Chapter 19, and we've gone through most of the topics between lab and lecture, so we need to finish platelets and we need to finish the cliff notes of clotting. And I say the cliff notes because this is a very complex process, and in fact, I still have to sometimes go back and watch the videos for med students to try and remember all the little intricate details of clotting. And if I have to go and watch videos to remember that, obviously we are not going to go to that level for our purposes. We're going to hit, like I said, the cliff notes so you are familiar with some terms. In the event you end up working for an internal medicine, you work for a hospitalist, or you work in ICU, again, cardiovascular disease is one of the most common causes of deaths, and many Americans are on some type of cardiovascular related medications, whether that's for their cholesterol, that's for their blood pressure, blood thinners. So you are probably, as a medical professional, going to run into people taking some medications related to their cardiovascular system. And a lot of the medications related to blood thinning are going to affect the platelets in the clotting process. All right. And so with that being, and then even just people like my parents, they take aspirin every day. You know, part of that comes back to, again, aspirin and how it affects some of the clotting factors in the clotting process. You don't have to know those details, because if and when you end up getting into the medical world to that level, then yes, you can come and revisit the clotting process and come and revisit every little intricate protein and detail. If you end up going into research and you end up working for pharmaceuticals that sell, blood thinners or blood clotting type drugs, then you're going to also probably get into more of the details, and you'll be able to come back and explain it to me and help me remember. But in all of these processes, we're going to stick to the cliff notes. You don't have to know every detail, and knowing every detail, that would be great, but that's like PhD level work or pharmaceutical level work. So it's not something I'm going to hold you to having to know. So when you see slides with all of these factors and all of these proteins and all of these things listed, do not freak out. We're not expected to know all of that. Okay? All right, so the third of our formed elements are platelets. Platelets are going to be also known as thrombocytes. Platelets are going to be like red blood cells. They are not technically cells. They are fragments from cells, and the cell they came from was this big, huge cell that came eventually from that myeloid progenitor cell from that hemocytoblast. So through some different, again, cytokines, colony stimulating factors, and some interleukins helped with other factors to push the hematopoietic stem cell to become the myeloid cell. The myeloid cell, again, with a little cocktail, and then TPO probably pushed it to then start down the platelet pathway, and it became a megakaryocyte, and then it fragmented into these platelets. Okay. Now, platelets are a little bit more complex in some ways than red blood cells, because they still, as the picture shows, they still have some intracellular organelles. They have some glycogen, so they can make, again, glucose from glycogen, and then glucose can be broken down to pyruvate and can generate some ATP. They still have some endoplasmic reticulum. They still have some mitochondrial type organelles, so they can still make some big amounts of energy, ADP and ATP, and then they have other signaling molecules inside many of their vesicles that are going to be important for not only activating themselves and pulling in more platelets towards them, but also to, again, start like all the other cells, activating the surrounding area, activating cells in that area, and even trying to start signaling, hey, we just used a bunch of platelets, we might need to be replenished. Okay, so platelets only live for about 9 to 12, so about 2 weeks, okay? In the spleen, remember our macrophages live in the spleen, and our macrophages, as blood flows through, they bump into red blood cells. Well, they'll bump into platelets. Okay, stand by. Matthew, you have your meeting, okay? All right, and so they are going to, the macrophages, when they see the platelets. And again, don't ask me the details on how they know the red blood cells are old or how they know the platelets are old. They will be able to pull the platelets through phagocytosis inside. They'll digest it. They will take the phospholipids, the triglycerides, the fatty acids, the glycerol, the pieces and parts. Go do your work. You have a meeting with the library. All right, sorry. They'll do all of that stuff and try to make it reusable so it can be sent back to the bone marrow to be reconstituted into more red blood cells, white blood cells, or more platelets. Okay. So you have platelets circulating. Now, again, since most of your blood is sitting in your veins, many of your platelets are sitting in your veins. So that's why if you have a big injury, and again, when you think about trauma, sometimes you'll see on TVs or sometimes you'll see or hear in your volunteer or your time spent that they have to give people blood and they have to give people platelets because if they do another surgery, there might not be enough platelets to help with the clotting process for the damage they're going to cause to the body when they cut in and start messing around with things. All right. So you do have a lot of platelets, and they are some in your veins sitting in your reserve, but even those platelets, again, they have a high turnover rate. If you have some major catastrophic trauma from a car accident or something, you might have to wait on some days for the non-urgent surgeries to happen, like if somebody broke their arm or broke their leg or broke their hip, because they might need to try to let the platelets reaccumulate, let the body kind of settle, and then they might need to even give blood and give platelets into the system. If you want $1 million idea, if you can figure out again how to grow platelets from stem cells in a petri dish, take the platelets and freeze dry them so we can stick them in baggies in backpacks so our medics in the field and our medics in field hospitals can have platelets available, freeze dried to quickly throw into an IV and reconstitute and get into the system, or quickly somehow get them on the injury site and help with the clotting process, you will have millions and millions of dollars. So just like our blood donations, we have not figured out the perfect cocktail to make platelets outside of the body in the lab, and we haven't found a good way to store platelets. We haven't found a good way to harvest them. We haven't found a good way to make them a long shelf life. But if we could figure that out, you would have lots and lots and lots of money. Okay? All right, so the main thing I really want to talk about today is the clotting process. All right, so it's also known as hemostasis. Hemostasis is going to have 3 key steps. The first step is happening basically as the injury is happening, as the body is becoming aware that cells are exploding, and when cells explode, calcium, potassium, phosphate, adenine, DNA, RNA, ADP, GDP, UDP, all of those molecules that are, again, supposed to be inside the cell, inside the nucleus, or some of it inside the nucleus, but inside the cell membrane, all of a sudden they become part of the extracellular environment. And they're not supposed to be there, especially things like potassium and phosphate and the nucleotides in high amounts like adenine, guanine, and ADP and AMP and GDP, all right? So when those are all of a sudden part of your extracellular environment, that is going to do some triggering for osmosis to bring fluid to the area. That is going to do some triggering for cells in the area to know, hey, somebody just exploded, there's damage here. That's going to start cascade of signaling to bring immune cells to the area. And then there's going to be some of those molecules that are not supposed to be in high concentrations in our extracellular fluid, when all of a sudden they are, and they interact with endothelial cells, when they interact with smooth muscle cells, they are going to directly then trigger the smooth muscle in the arterioles in and around that area to constrict. Okay? So constriction is going to be the first thing we normally see in and while the injury is occurring. And part of the constriction is my blood is precious. I do not want to lose a significant amount of the blood volume because, again, I have 5 liters. I don't want to lose a good liter or 2 liters. That's not a good thing. So I don't want to lose the volume. And then more specifically within that volume, I don't want to lose the millions and millions and millions of red blood cells that I would lose in a few drops of blood, because that takes a long time for me to replenish. So the vascular spasm is trying to quickly minimize the amount of fluid, total blood loss, and then more specifically minimize how much of the red blood cells that it takes me a few days to weeks to make. I want to minimize the loss. Okay, so the vascular spasm, also known as the vasoconstriction phase, is going to be our first phase, and it's happening sometimes instantaneous to cells being ripped open by the injury happening. Okay. Other things that are going to start to cause that vasoconstriction are going to be, again, endothelial cells that are lining those simple squamous cells. They're going to know that an injury is happening, and they're going to start to release, again, signals to the smooth muscle in the arterioles nearby and around them to constrict. All right. And that's going to help us constrict. And then even our nervous system, when it starts to get pain signals, when it starts to get maybe squeezed because of fluid accumulation, because fluid from the blood and fluid through osmosis with all these extra solutes being brought and introduced into the extracellular environment, is going to maybe even trigger some release of some chemicals to cause vascular muscle constriction. So the vascular spasm is going to be happening pretty quickly. I mean, this is happening as the injury is happening and then in that immediate time after. And the goal is to minimize the fluid loss, minimize the red blood cell loss, and then try to, again, constrict and divert blood to the vessels that are not damaged. Okay? The second phase is going to be where, okay, we have this vessel that has an opening, that has a point that the stuff is leaking out. We need to plug it. And the initial plug is going to be with our platelets. Now remember, platelets are soluble in water, and we can put them and try to kind of like put a patch on, but the patch is still potentially going to let leak out some of the fluid and some of the solutes because it's not a perfect patch. All right. If the injury is small enough, it might be enough of a patch that the vessel and the endothelial cells can quickly try to go through mitosis and reestablish and fix. But most of the time, if it's a significant damage, the platelet plug is going to at least protect you from losing vast amounts of fluid and your red blood cell. It's not maybe going to solve everything entirely. All right. So then the third phase is when we start to make a plug that has a gel and a insoluble component that starts to make the barrier to keep blood in and to keep extracellular fluid out. And that's when we add in the fibrin, which comes from our fibrinogen proteins, which comes and is basically made from fibrinogen, gets a little snipping, and that happens because a cascade of events happens between the vessel being injured, the platelets being activated, chemicals being released by endothelial cells, by the area, and we get a bunch of little ants starting to turn enzymes, and it's a snowball effect, and more of these enzymes are then turned on, and they all turn on fibrinogen to be converted to fibrin. Okay? So just to, again, break it down, in vasoconstriction, you are going to have smooth muscle contraction. That smooth muscle contraction can come from some of the endothelial cells. So if these healthy, simple squamous cells that are not injured, but they start to be aware of the environment that's full of inappropriate things like large amounts of potassium and ADH, or ADP, TTP, CTP, phosphate, and things that are not supposed to be in excess in the extracellular fluid. You might also see, again, some nerves in this area release, and then some, again, just natural that cell exploded, and all these things like potassium and ADP and GDP that are not supposed to be in the extracellular can also directly affect the smooth muscle to contract. All right. As the smooth muscle is contracting on the healthy or the messed up vessel, some of the molecules that are also released by the endothelial cells, by the explosion of a cell, by the surrounding cells that are aware now that an injury has happened, they are going to start to release against signaling messengers that are going to tell other vessels in the area to dilate and are going to help draw neutrophils and help draw macrophages to this area so they can start, again, spitting on the environment and cleaning up debris so they can start ingesting it. Okay? The second phase is going to be where we start to let the platelets get plugged, all right? And the platelets are going to start to get activated because they're going to be told to activate by some of the endothelial cells that are, again, going to start to secrete some molecules to tell the platelets to adhere to each other. And then that'll also trigger the platelets to start to release molecules like ADP and TXA2. And again, you don't have to know those molecules. And those molecules will start to tell the platelets to attract to each other. And then if they come into contact with things like collagen and elastin protein, that'll start to, again, activate them. So the platelets will start to, again, form this temporary plug. And remember, the platelets are still mostly water soluble. So I could still have leaking out there a little bit more of my plasma fluid and my sodium and my potassium and bicarbonate, and my small molecules that are leaking into this interstitial area. And so normally when we think of injuries, we also think of it's going to swell up. And that swelling is a combination of, again, changes in osmosis with my cells that blew up, changes in osmosis as some of my liquid from that injury area are becoming more part of the cellular area and causing more osmosis and water to accumulate there. And then remember, my white blood cells are coming into this area and they're spitting on and releasing chemicals, and that's also drawing water and fluid and more attention to this area. All right, so there's lots of swelling happening. And that swelling would be normal because of all the changes happening to solute and volume and cell being attracted to this area. All right. And then again, the final phase of clotting is I need to, and you probably can't see it, but there's these little blue lines that represents fibrinogen. And I want to get fibrinogen to make this little meshwork around the platelets and in between the platelets, because once that happens, fibrin is insoluble in water. So it'll kind of buff away water and anything suspended in my plasma to stay, that's in the blood, stay and be a part of the blood. And it'll also help, again, push away water and solutes that are outside of the blood vessel. So again, that's part of the interstitial environment to stay part of the interstitial environment. And so it also helps me with that. All right. Now, with the phase of getting fibrin and getting not just one or two fibrin molecules cut up, and joined together, but to get a crap load of fibrinogen converted into fibrin, and to do that quickly, I am going to use my intrinsic and my extrinsic pathway. So think of it as I am going to use not just me working, but I'm going to recruit two friends, and then they're going to recruit more friends and more friends, and together we are all going to be snipping fibrinogen into fibrin. And the way I recruit more and more friends is I might email some friends and I might call some friends, but I'm going to use email and calls to try to get more of my molecules or more of my enzymes going that eventually lead to fibrinogen being cut into fibrin. Okay, so the intrinsic pathway is going to be something inside the blood vessel, inside the blood itself, is triggering, again, the cascade of events that lead to eventually factor 10 being turned on. So factor 10 is a protein that normally does nothing, and then when it gets turned on, it's going to start to initiate some enzymes that lead to fibrinogen being cut up into fibrin pieces. Okay. Now, Extrinsic pathway is going to be initiated from external factors outside of the blood. So this is more of like the endothelial cells and the underlying cells in the tissue start to generate, again, some molecules that lead to factor 10 being turned on. Okay, do you need to know those factors that are part of the intrinsic and extrinsic pathway? No. What you do, one little fact you do need to know is calcium is important to get some of those molecules turned on. So calcium is a huge component of the intrinsic and extrinsic pathways to get them working. So if somebody was hugely calcium deficient, they might be slow clotters because if calcium's not around, these molecules cannot turn on. Okay, and the key part of the intrinsic and extrinsic pathway is both of them lead to turning on a lot of factor 10 or factor X, because 10 is X in Roman numerals. And so again, think of it as I email a bunch of people to turn on factor 10. I call a bunch of people, but the end result is I maybe activate by myself 5 molecules of factor 10, but if I'm emailing and I'm calling and 5 people then also activate 5 factor 10s, now across the board, I'm at like 100 factor 10s are activated. And if 100 factor 10s are activated, they can quickly turn 100 to 1,000 molecules of prothrombin into thrombin, and thrombin then starts to do on fibrinogen, and I generate very quickly lots and lots and lots of little fibrin particles that want to all join together into a strand and then form a meshwork that over and in between the platelets starts to make the plug insoluble and starts to help separate that plug so it becomes part of the vessel wall and it's no longer kind of immersed into the blood and it's no longer really merged or immersed and part of the interstitial system. Okay. Now, one of the big regulation pieces is I want all of this to happen quickly and I want it to snowball and kind of like a feedback loop really quickly lead to a lot of platelets and a lot of fibrin, and I want it to happen in that injury place, but I don't want it to happen outside of the injury spot. Okay, and so how do I keep it from snowballing outward and becoming part of the area that is not damaged? Well, those endothelial cells, so looking over here, these endothelial cells that are not damaged, part of what their job to do is to create factors, create proteins, create cytokines that on their membrane are going to try to prevent platelets from aggregating, prevent platelets from activating, and prevent fibrinogen and fibrin from becoming part of the attached to them. All right, so they play a key role in that. Other reasons why people sometimes take anticoagulants or blood thinners or drugs that try to prevent the clotting process is one of the ways you can have a lot of damage, and a lot of damage to your endothelial cells can lead to the clotting process to start. That extrinsic pathway comes from high blood pressure. And again, high blood pressure, that high force, that high stress and friction happening as blood moves through your vessels. And so people with high blood pressure, again, naturally as blood flows through, they are rubbing and friction and damaging our endothelial cells, which can lead to the extrinsic pathway. So that's one of the reasons why people with high blood pressure, eventually, if their blood pressure is not going to really come down to an acceptable place on drugs, they start having to add a cocktail to their regimen of, oh, I need to take some blood thinners and I need to take something that prevents the extrinsic pathway from getting started and ramped up, so I don't have clots being thrown and potentially throwing a clot that could be big enough to then stroke out my brain or stroke out some important place in my lungs. Okay. Now, once I make a plug, all right, another part of some of the cytokines that the platelets are releasing and the cytokines that my cells are releasing that are, drawing in immune cells and are trying to signal, hey, we got damage, and immune cells are throwing in their chemicals. Some of those chemicals are going to also try to trigger cells in and around this healthy tissue area to start to go through mitosis. And why would I want them to eventually go through mitosis? That might take a few days, but eventually I need these endothelial cells to grow and replace where the clot is. And I need these smooth muscle cells to grow and replace and and take up the space where the clot is and regrow my muscle layer to a smooth layer, to regrow my connective tissue, replant collagen and elastin fibers in that connective tissue area, and regrow my endothelial cells so I have again a line of endothelial cells throughout my entire cardiovascular system. So some of those factors that are being released in all this process are also growth factors, right? And they're going to take a few days to get the cells to go through, because they have to DNA replicate, then they have to go through mitosis, and then they have to kind of after mitosis grow and expand and move into that place that is currently occupied by the clot. So I'm also going to need to get rid of the clot as that's happening. And part of getting rid of the clot is I got to get rid of the fibrin and I got to get rid of the platelets, all right? And the way to get rid of fibrin, because it's an insoluble, non-water-loving molecule, is I'm going to have to activate plasmin, and plasmin comes from the molecule plasminogen, okay? And one of the molecules, this is not a great slide because it moved up, but TPA, and you'll hear about if someone has a stroke, if you can get them to the hospital within 2 hours and they can give them TPA. TPA is one of the molecules that's naturally made by some of our cells during the clotting process to start to initiate quickly in a day or two that process of removing the clot, okay, and breaking down the fibrin. All right, and so TPA eventually leads to plasminogen being converted to plasmin, and plasmin then eats fibrin and removes it from a clot. So it's how we get rid of the clot. All right, well, one of the reasons why we, especially those suffering a stroke, if they can get to a hospital within 2 hours, TPA, is if I can start the fibrinolysis process before the stroke causes occlusions and complete loss of blood supply to cells, and then those cells go through death, and then you don't get them back. If I can start getting that clot to be broken up, so some blood maybe scoots through and some oxygen and nutrients get to the cells to keep them alive, I am going to have a better outcome with my stroke patient. All right. And why is 2 hours the big window? Because if you are going to see cell death, it's going to happen probably if the clot is able to lodge itself for 2 hours or more. Okay. And why is that a big deal? Well, what's happening in our rural communities is that hospitals and emergency rooms are going away. And so for some rural areas, it might be a 2-hour drive to the nearest urgent kind of trauma, stroke, cardiovascular hospital. And so that's part of the reason why if someone thinks they're having a stroke or starts exhibiting stroke symptoms, they might need to get on the road quickly or call the ambulance and hope that they get there within and get them back to the doctor within that 2-hour window. All right. So for some of you that might move into some rural areas, rural country living might be wonderful until you get old and you start having this risk of throwing a clot and making a stroke happen. All right. And you can't then get TPA and this treatment. And so your outcome for maybe how much of recovery with maybe the right side paralysis and loss of speech and loss of function you have might be more permanent because you didn't get that TPA treatment within that 2-hour window. Okay, I think that is all I have. So I'm going to stop the recording.